Today we're going to do enzyme regulation. So, let's see. Eeny, meeny, miny, Ben. How would you define regulation? Okay, glad I asked. Regulation is about a limit or its rules to follow. So when you have regulations, it's basically going to refer to limits or a system of control. Does anybody here go fishing? Has anybody ever gone fishing at least? Okay, you've gone fishing, perfect. Has anyone ever eaten stone crabs before? All right, cool. So this is going to work for both of you. So let's say that Alexa goes fishing and she catches a snook. And this snook is about 2.5 feet long. This is legal. Alexa can keep that snook. She can take it home and cook it and have dinner. But let's say that Alexa's fishing buddy catches a half-foot snook. That is illegal to catch. That's a juvenile. Why would the powers that be with the state of Florida government, Florida Fish and Wildlife, why would they make it illegal to catch a baby or juvenile snook? What do you think? Think about nature. Think about succession. What hasn't this juvenile snook done yet? It hasn't reproduced. And so what would happen to a population if you were to remove all the pre-reproductive individuals, the kids that haven't reproduced yet? What would happen to the population in the not too distant future? It would decrease and maybe get wiped out. So this is why it would be illegal to capture and keep juvenile fish. They haven't reproduced yet. This 2.5-foot snook, when it's gotten to that size, it has likely reproduced. So it's done its contribution to the population. You can keep it. There are regulations on how big a particular species of fish you can get, or what time of the year that you can catch the fish. Sometimes some species are a catch and release only. Now, another thing, I mentioned stone crabs. Tasty. Well, just like any other crab, they have two pinchers, two claws. When you're stone crabbing out in the Gulf of Mexico or somewhere, how many claws are you allowed to take per crab? One. Why? You catch a crab, you take off one claw, and you let it go. It will grow back, but that's not the reason why. Why are you only allowed to take one? Because by that, if it grows back, then why not take both? They'll grow back. Zoe? Oh, wrong one. Go ahead, Bill. They needed to survive. They needed to survive, and also they needed to defend themselves. They need one claw to feed themselves. So you cut one claw off, it can defend itself, it can feed itself. And then when you go back next year, the claw has grown back. And so you catch that same crab, you take the claw off, and the crab by that time is just like, dude, really? Well, this is another regulation that we have in Florida where you're only allowed to take one claw. It's the same thing with the fish. If you continue to take both claws off, the population is going to get dwindled down to probably nothing, and there'll be no more stone crabs. I see people going up and down Bayshore Boulevard. I know you see with a big net, and they'll try to catch all the blue crabs along the seawall. If you kept doing that, there will be no more blue crabs. You gotta let them regenerate. You have to let them repopulate. In fact, there are some areas of the Florida Gulf Coast, I think in Louisiana, where oysters, maybe it's Louisiana or somewhere, northern Florida, the Panhandle area, oysters have been collected to nearly wipe. So much has been collected that that whole area that I'm thinking of, I think it's in the Panhandle, has been wiped out. And so the city or the county said, no more catching oysters for 5 years. Nothing. You have to let them recover. You have to let them come back because they didn't have very strong regulation, and people just went crazy and they took as much as they could. So sometimes regulations are necessary to keep you from taking too much. Well, when it comes to enzyme regulation, an enzyme in substrate can often make too much product. And when you have excessive product, you need to shut it down. Let's say that you go into your pantry and you have 20 boxes of Cheerios. Your mom is getting ready to go to the grocery store. Honey, I'm going to the store. I'm going to get some more cereal. Like, Mom, no, we're good. We have 20 boxes. Don't get any more. On the other hand, let's say you don't have any cereal. You say, Mom, can you get some cereal? We're out. So we want to stay within that range of not enough to too much, right in the middle. We want to stay at a very healthy equilibrium where we're just the right amount. We don't have too much, we don't have too little, just right. And that's what enzyme regulation does. So in enzyme regulation, the amount of product synthesized by enzyme substrate complexes is regulated. And regulation is just another word for controlled or is limited. All right, so there's going to be several factors that I'm going to introduce to you today that helps to regulate the amount of enzyme that can actually be produced. And the first one that we're going to do today is going to be co-enzymes and co-factors. I think there's 1, 2, 3, there's 4 tools of regulation we'll go over today. This is the first one. So, co-enzymes and cofactors are used up in a reaction, unlike enzymes. So as I mentioned yesterday, and when you have a chemical reaction, enzymes are not used up. You can use them over and over and over and over and over and over and over again. Coenzymes and cofactors can be used up. So what do they do? Coenzymes and cofactors aid an enzyme in forming a compatible active site for a substrate. So that's a great little description there, but I think a picture speaks 1,000 words. So let's draw an actual diagram of what a coenzyme, an enzyme, and a substrate look like and how they interact with each other, shall we? Here we go. This structure is our enzyme. This structure is our substrate. What do you call the place where the substrate meets the active site? What is? Darn it! I just said it. That's the active site. I meant to say enzyme. There's the active site. Now, what's wrong with this picture, Mitchell? Can that substrate fit into that active site? No. So here's what we're missing. I haven't drawn it yet. Hopefully you all can deduce, as Mitchell just did, that substrate cannot fit in that active site. So you cannot make product right now. It's not going to happen. No way. Unless you get yourself one of these. This, ladies and gentlemen, is what we call a coenzyme. You can probably see how this is going to work. The coenzyme can fit into the active site to make the active site more compatible for a substrate. So let's draw them together. Wrong color. Okay, you can now form an enzyme substrate complex. And what is formed when an enzyme and a substrate form an enzyme substrate complex? What do you make? You guys recall? This will be the first of 4 times I do this today. So don't leave me hanging on times, 2 through 4. You make products. What do you make? All right. I'm going to do that a lot today. Now, look at how I draw this real fast. Check this out. Someone earlier today said, how come you didn't make it green? I'm really hanging on every word you're saying. Because is it a substrate anymore? No, it's not. It's a product. The products are totally different than what the substrate was. And once that coenzyme leaves that active site, you cannot make any more products. So let's say that you make enough product to get up to maybe a few micrograms. And once you've reached your quota, the coenzyme is going to detach and the active site is going to be incompatible again. Any questions on coenzymes and cofactors and how they work? I want you to really pay attention to how this is regulated. This is controlling how much product is made. Yes? Cofactor does the same thing. I'm not going to do both. I've just been doing the same thing twice. It's the same function. Yeah. Yeah. Ugh. All right. So those are coenzymes and cofactors. Let's move on to the next item. Number 2 is going to be competitive inhibition. During your test last week, a few students saw the word inhibit on the test. They said, Mr. Thorson, what does inhibit mean, or inhibition? Inhibition means to stop, or to slow down, or to stop. If you want to hear it used in a sentence, as a civilization, we want to inhibit the spread of coronavirus. You want to stop it. You want to slow it down. So, how does this work? Well, you all know that an active site is where a substrate can bind to an enzyme. Well, in this case, a substrate and an inhibitor. And there's types of inhibitors. We're going to call this a competitive inhibitor. Competes for the active sites of an enzyme. So I like to think of this as musical chairs. If you haven't played musical chairs in your life, you haven't had a childhood. Your childhood is sad. So you've got the chair right here, playing the music. You guys know how it goes. You're like, okay, you got a dancer on the chair. You run around here so you can be the first one to sit. And you keep on dancing. You get kind of nervous. Oh my gosh, when's the music gonna stop? Music stop. Run for it. Someone just lost. Not you. You're busting the seat. The seat is the active site. One person is the inhibitor. The other person is the substrate. So, what happens if the substrate wins, quote unquote? Product is formed. What if the inhibitor wins? Anybody want to take a guess? Ladies? Anyone? No product. Basically, the enzyme is shut down. So I'm going to draw both scenarios for you, all right? All right, here we go. This green molecule is your substrate. Here's our new character, everybody. This is the inhibitor. How many places are there for the substrate and the inhibitor? One each or one for both? I heard both. All right, both answers. How many active sites do you see on the enzyme? How many different shapes do you see? How many different structures do you see could fit in that active site? There's two people, there's one seat. You can't have two of them share the seat. So what I want to do is I want to simulate what would happen in one direction if the substrate won, quote unquote, and then what would happen if the inhibitor won. Okay, so as I asked you earlier, what happens when you get a substrate and an enzyme? What do you make? No, not yet. When an enzyme and a substrate come together, what do you call that? Enzyme substrate complex. And what does an enzyme substrate complex make? There you go. So That's the second time we've done that this period. We're going to do it 2 more times. I think 2 more times. Yep, 2 more times. Now, let's see what happens when you do the inhibitor. The inhibitor is occupied the active sites. No reaction. All right. Yes. Products are things, did you ever take chemistry? Well, in chemistry, you have, check it out. You have reactants, and reactants will react together to form products. So you took freshman bio, right? So in photosynthesis, you have 6 molecules of carbon dioxide, you have 6 molecules of water. They react with sunlight to form 1 molecule of glucose and 6 molecules of oxygen. These are your reactants. In the event of an enzyme in a substrate, this is what the substrate would be. When they go through the reaction, they form something new. That's what a chemical reaction is all about. You take your reactants, you rearrange them, and you form something brand new called your products. There's something new. It could be a smaller metabolized protein that might have to go perform some function somewhere. You want to break down a substrate or build up a substrate into a new type of molecule for, and whether it be something structural or something functional. And so these are reactants. These are products. Now, a substrate, everybody, will eventually become product. It just is going to take a long time. But thanks to enzymes, it makes it happen much, 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 much faster. Now, when there's no reaction, it's because the inhibitor is occupying the active sites. The inhibitor occupies the active site, basically budding out the substrate. Yeah. Okay, well, you have inhibitors because there are times, and not just eukaryotic organisms, but prokaryotic organisms, where you are making too much product, like maybe you're making too much, maybe you have a piece of food, maybe a substrate, like a disaccharide is a food molecule. You need to break it down into its monosaccharides in order for it to get into your bloodstream. So you have enzymes to break that down. Well, there's also cases like maybe neurotransmitters, maybe other types of proteins that you want to limit how much you have. And so inhibitors are going to prevent you from making too much of it. One example I think of in bacteria is lipase, or not lipase, lactase. Lactase breaks down milk. Do you want to make lactase if there is no lactose in the area? No. The purpose of the enzyme lactase is to break down the sugar lactose. Lactose is in milk. If there is no lactose in the environment, like inside the bacteria, there is no point on making lactase. So you want to plug in an inhibitor into, in this case, the gene that makes the enzyme. So you are not making something that you don't necessarily need at that time. So there's inhibitor, and we also have things like pharmaceutical drugs that serve as inhibitors or activators to try to regulate maybe some maybe psychological condition, maybe a pathological condition you might have. For instance, let's say you have a virus. Check this out. You have a virus, and my hand right here, my open hand, is serving as a cell receptor. This phone is part of the virus. The way the virus gets into your cells is it has to fit inside your receptor. It does it like this. Once the virus is bonded with your receptor, it's able to get into your cells. That's bad. You'll follow me so far? So let's say that you take a drug from a clinic, where this is what you take. You take it orally, you take it intravenously, whatever. And this is going to go into your receptor and basically block the virus from getting in. That's what inhibitors do. A lot of times we make medicines that are based on the whole nature of competitive inhibition or non-competitive inhibition. Yeah.
So when the inhibitor is attached to the enzyme, it's not going to ever come out.
It will, it can. It just depends on if you need that, if you are ready to make more of that product. Remember, there's such thing as too much. You want to find a nice equilibrium. If you're within that range where it's good to be making it, then you're going to make it. The inhibitor will come off. There's different triggers like pH level, hydrophobic, hydrophilic, chemical signals, what have you. So let's say you have too much product, the inhibitor is going to plug in to stop you from making it. Let's say that you don't have enough. The inhibitor is going to let go so you can start making more. All right.
So some people don't have enough inhibitors in their body, which is why they have too much
Yeah, there's things like that, and yeah, then you have to have some sort of therapy. Yes.
So if we need more, if we need something to be inhibited, there are like signals that sent up to our brain, and there's like a negative.
Well, it's not just your brain; it's signals that are sent to your DNA, and your DNA has all the codes to make those enzymes and to make those inhibitors. And so your DNA will be transcribed and translated into a RNA and into a protein. And then that will go, remember I was saying whenever a protein was made, it goes off to do whatever it is it's supposed to do. Transcription is where you take a DNA code and you translate it into a protein. So transcription is DNA to RNA, translation is RNA to protein. You want to take the code from DNA, it's not your brain, it's just your genetics. You take the code written in the DNA, you make an RNA segment. The RNA leaves the nucleus, it goes into the cytoplasm, and makes a protein with a ribosome. That protein isn't going to do whatever it's supposed to do, and that protein might be an enzyme, it might be an activator, it might be an inhibitor. And so these signals they're sent through hormones, they're sent through chemical messages. It's all it's all about that homeostatic condition that we want to maintain so we don't get too off balance biochemically. It's not very easy to explain. It's not very easy to understand. But I will tell you, when we get through Unit 5, that's where it really comes together, because that's molecular genetics. Yes?
So basically, like, an outside force sends a signal to a cell, and the cell goes, and that signal goes to the DNA, and the DNA translates the RNA, and then transcribes the protein.
Yep, more or less. All right. Okay. The next one I want to do is going to be non-competitive inhibition. Okay, well, the example I gave you guys for competitive inhibition was musical chairs, 2 people, 1 seat. In competitive inhibition, there's 2 seats, but this is a weird analogy. 1 person in 1 seat sabotages the other seat. So let's say that, I'm playing musical chairs by myself. You're gonna sit there. I'm dancing around this chair, and right when I was about to sit down for the chair, oh no, I fall on my butt. What he just did is he took away my active sites. And this is an entirely new section of enzyme that I haven't explained to you yet. We're going to get into it now. It is called the allosteric sites. So notice that the reason this is called non-competitive is did he and I go for the same seat? We did not. We did not directly compete. But he still took away my chair. So he inhibited my ability to sit down. In biological terms, he inhibited my active site so I couldn't make a complex. So, in non-competitive inhibition, a molecule called an inhibitor binds to a site that is not the active site. This union changes the active site of the enzyme to be incompatible with the substrate. So what I need you to gather from this title, non-competitive, is that they are not directly going up against each other, the inhibitor and the substrate. Instead, the inhibitor is going to take away the active site from the substrate in a different way. So, guys, this alternative site I mentioned, I don't know what it is about this word. A lot of kids forget it. I don't know why. It is called the allosteric site. I devote a little extra brain power to that. I say that just because in the past, a lot of people forget. I don't know why. Allosteric. A-L-L-O-S-T-E-R-I-C. Allosteric. So this is known as allosteric regulation. So here we go. This is our enzyme. That open curved surface is the active site. This little squared shape opening is going to be the allosteric site. Okay. This green molecule is going to serve as our substrate. And this red molecule I just drew, this little square, is going to serve as our inhibitor. Now, remember that to inhibit means to stop or to slow down. So after you guys get this drawn, I'm going to show you what happens if the inhibitor doesn't do what it's supposed to do, and then I'm going to show you what happens when the inhibitor does do what it's supposed to do. It should be pretty self-explanatory what would happen if the inhibitor does not do what it's supposed to do. The enzyme is going to have no problems bonding with the substrate. What do you call it where an enzyme bonds with the substrate? A what? Enzyme substrate complex. And what does an enzyme substrate complex form? Products, right. Okay, it's the third time that that's been drawn today. Hopefully it's starting to sink in. enzyme and substrate come together at the active site of the enzyme. They form an enzyme substrate complex, and that is going to catalyze the reaction to basically allow the substrate to react to form product or products, whatever word you use. Yes, Mark?
After it makes a product, is the enzyme like right away ready to make a new
Yep, it is not used up in the reaction. It can be used over and over and over. Yes.
So is the inhibitor just kind of like hanging out around?
Sometimes it's not around. What I'm just doing in this situation is just showing you both of them, okay? So now what I want you guys to see is what happens if the inhibitor plugs into that allosteric site. So here's something new. So right here, the inhibitor is fit into the allosteric site. Check this out. You ready? Watch. What happened to the active site? It changed. Can this substrate fit into the active site? Nope. So what's going to happen with product? There is none. So this is going to result in no reaction. The reason this results in no reaction is the inhibitor binding to the allosteric site has altered the active site and thus there can be no enzyme substrate complex. And if there cannot be an enzyme substrate complex, there cannot be product. Yeah.
How are they bonding together? What's holding them together?
It's like the way that enzymes and inhibitors and active sites and substrates work is it's just a lock and a key. They fit together. They're meant for each other. That's what your DNA is there for. Your DNA has the codes to make these different molecules.
What determines what draws an enzyme into the...
It could be attractive forces. It could be the type of bonding, hydrogen bonding, intermolecular forces, partial positive, partial negative, electrostatic attraction.
So like the electro attraction, whatever, is stronger with substrate, and the substrate's going to go inside the...
Yeah.
And then same thing for inhibitors.
Yeah, that can work. All right, we have one more to get to. We got to kind of hustle. Class is over in about 5 minutes. What I do want to make a note of, guys, and this is going to save us time, non-competitive inhibition, non-competitive inhibition can also be considered to be allosteric inhibition. It's generally considered the same concept. So we don't have to do this twice. So the opposite of allosteric inhibition will be the last one that we do today. It is going to be called allosteric activation. In allosteric activation, an enzyme's active site is not compatible with a substrate. The allosteric activator binds with the allosteric site changing the active site to be more compatible with the substrate. Keep spelling that wrong. Okay, so I got to draw this quick. Oops, I did that already. I did it wrong. All right, so this is our enzyme. This is the allosteric site. This is the active site. Look how weird it looks. Especially if I told you that is your substrate. Look at the shape of the substrate. Definitely not a compatible match. And there is your allosteric activator. So what the activator basically does is it turns the enzyme on in this case. So as you guys can see in this image, the activator causes a change in the active site. This allows the substrate to plug in. This is going to form our enzyme substrate complex. And last time, what do you get when an enzyme and a substrate, enzyme substrate complex come together? What do you make? There you go.
Does it go back to its original
What's it?
structure? Does the enzyme go back to its original structure?
Once, well, if the activator comes out of it, it will. But as long as the activator stays in there, it's gonna be changed to an altered, more compatible form. But as soon as that activator detaches from the allosteric site, it will go back to that incompatible active site.
And would it be detached from one of those signals that we were talking
Yeah.
about?
Yep. All right. That's all.